I will introduce briefly the IMCAS and the CGMCC in China. And I think the ACM we can meet every year. That is very important because in these recent years, I think the whole world meet many difficulties. For example, the pandemic of COVID-19 evenly maybe nearly stops the globalization. And scientists focus on the microbe study should be communication more and more and to make this world better again. And this is my topic. I will briefly introduce the IMCAS. Most of our friends has visited Beijing and into the IMCAS, but maybe some of our new participants do not come to our institute. So welcome if there are future opportunity, welcome to Beijing and visit us. And secondly, I will introduce our reforms recently on the CGMCC. And I will also mention our future development directions and communicate with all of our participants this afternoon. And IMCAS, today is the 30th anniversary of the NAS. But IMCAS is founded about 67 years ago. That is in 1958. And we are maybe, I guess, maybe IMCAS is one of the largest government-sponsored institute to study microbiology in this world, because we covered almost all research aspects of the microbiology. As you know, from the isolation and the taxonomy of the microbes to the evolutionary biology and the ecology, molecular biology, biochemistry, we even have the plant pathology and also the synthetic biology and the pathogen and the immunology. So we covered a wide range of studies in biological sciences. And secondly, now we almost have 96 principal investigators. So we have 1,000 of staffs and scientists and 1,000 of graduate students. About nearly 400 are PhD students. So in recent years, because Chinese government have change and reforms and revolution in our scientific systems. So before we have 3 state K-lab, but now we combined these 3 K-lab. One is the K-lab for microbial resource, and the second one is the state K-lab of mycology, and the third one is the state K-lab of plant genomics, mainly focused on the pathogen and the plant interaction. We combine these 3 state K-lab into one. This one is called state K-lab of microdiversity and innovation utilization. So this state K-lab is will use the microbial resources as its most important research materials and to have innovative utilization for the future study and the future development of biotechnology. This slide is not renewed because I renewed it in afternoon, but I will report in that version. And our institute also have several platforms for the study of microbes. And in the morning, Professor Juncai Ma have introduced the World Data Center for Microbe. It is also the National Scientific Data Center for Microbiology. And we also have a very large herbarium for the fungi. And this herbarium now is led by the Professor Lei Cai. And this is a wonderful place to support our mycology study. I will mainly introduce the CGMCC. CGMCC is is the most oldest stock center in China. It's funded by Professor Xin Fan. Even the history is longer than the IMCAS. So, this is our brief history of the CGMCC. CGMCC is established before the liberation of the People's Republic of China. It is established in the nearly 1960s. And after the PR China have established and the establish of our institute, the CGMCC is our most important platform to support our study in the institute. And in the recent, it is also one of the stock center to storage the microbes that is used for apply for the patents in China. And also, it is our national, one of our national microbial resource center. And this is our current status of the CGMCC. It's about collection is nearly 110,000 of strains. And we are now the secondary large stock center for the patent deposit. The first one is ATCC in USA. We believe maybe within the future 5 years will be a #1 because our increase rate is very high. And we also, all of these strains support the biotechnology development and also in the support of a basic study in China. And I will introduce, because in the 2 years ago, we reformed our management system of the CGMCC. Professor Yu Guanzhou is an old friend of most of our participants. After he retired, we appoint Ms. Ya Jinyu, also participant of this meeting, as the acting director. But the CGMCC is under the guidance and the leadership of our institute. So, begin the 2 years, we have management, we call it the smart management of microbial resources. The first one is we collect the very diverse of microbial resource as quickly as we can and as large as we can. For example, in these years, we collect most bacteria from the soil, especially in the northern east of China, and from the human being, and also from the animals. So we enlarge the biodiversity of our collection in these 2 years. And secondly, we innovate our storage skills and want to use a high throughput screen and identification method to enlarge our collection. And the third one, we enlarge and reinforce our global cooperation and standardization, including attend the meeting of ACM. And the smart management, just as the slide you show, we, besides of the general microbial resources, for recent years, we are more focused on the professional collections, including in the medical science. For example, including the virus, such as the SARS-CoV-2, because we have the ability to storage some of part of the pathogens. And also in the agriculture, for example, we set up a collection of the fish, because you know the disease anti-drug resistance bacteria or fungi are more and more. So besides of the antibiotics, phage might be one of the future choice to defend the drug-resistant bacteria or fungi. This is one of our reform. And we also set up a platform for the high-throughput identification by the single cell of the microbes. But you know, it is quite difficult because culture of pure culture depends not only on the species, it also depends on the relationship between the microbes or microbe and the host, or even quorum sensing have a very significant influence on the pure culture of the uncultured microbes. And we also want to connect more microbial resources to the biotechnology development. Maybe I will introduce some of the cases in the following slide. So in a brief summary, under the guidance of Ms. Yu as our new director, we reinforced our whole genome sequencing. As you know, we have a GCM 3-point version. And now we have sequenced thousands of type strings in our collection. We also set up high-throughput analysis of metabolic phenotypes, especially the drugs or the chemicals. And we promote our technology advancements in the preservation, as a professor have introduced in the morning. And one of our most goal is culture that uncountable microbes, because we do not know how many microbes are uncultured. Usually we say 70% to 90% of microbes are you cannot culture, but they are really important. And we also reinforce our collaboration network within China and with our international friendship. I will introduce some of our applications in the biotechnology or the biomanufacturing that is supported by the microbial resources. And every year, IMCAS select a 10 research result as our top 10 in that year. As you can see, in our institute, although these top 10 studies come from diverse areas of scientific research, but almost all of them are based on the microbial resources. This is what we want to do, because you know, in China, even the Chinese Society of Microbiology is located in the IMCAS, and we also have a CGMCC as a very important biological resources. So this is just the top 10 research in the last year. And besides, I mentioned our progress in the synthetic biology. I will firstly mention some of our research on the medical science. This is not renewed in this version. For example, during the SARS-CoV-2 pandemic, IMCAS, because we have the ability and we have established a P3 level laboratory which has certification to manipulate the HIV and the SARS-CoV-2. So we designed a recombinant protein vaccines. That is the first protein vaccines that is allowed for the clinical application. That vaccines has delivered nearly 3,000, to say 0.3 billion doses in this world. And we also designed antibody to cure the SARS-CoV-2. Our very short story is in the year of 2020, you know, President Trump, he is also the president now in USA. He also was infected by the SARS-CoV-2. And in that time, we the antibody that is designed by our institute, and we cooperate with the Lilly Corporation, has provided that antibody to the White House. But you know, President Trump did not select that antibody to cure himself because that is made in China. Yeah. So that is two cases that we, based on the microbial resources to development the vaccines and the antibodies. And this is a case from the synthetic biology. As you know, we can use synthetic biology, can use microbe to produce everything of the chemicals. But usually, if you want to use the patent to transfer to the industry section, are usually not successful. Take the vitamin B5 as an example. Many universities and research groups know how to use E. coli to produce the vitamin B5. But if you want to transfer your patent to the industrial sector, why you have to be valued? This is because the purification is very important, because we know during the purifications there is 3 byproducts that is produced together with vitamin B5. How to solve this question? Because if you have the 3 byproducts, the purification will cost more. So based on the calculation based on the large data from microbial resources, we eventually solve this problem. And this is the first product line in this world to produce vitamin B5. That is quite near the Korea. That is northern eastern of China, of the Harbin. We transfer this technology to the corporations. And this is one of our examples in the agriculture, modern, because this is in the north of the Korea. And this example show you, because you know there is a fungi, pathogenic fungi to infect the cotton, that is Bacillium. And there is no cultivar of cotton can resistance to this pathogenic fungi. Professor Huishan Guo in our institute have a basic research finding. She found that the small RNA can be transferred cross-kingdomly from the cotton to the pathogenic fungi. The paper is published on the Nature Plant. And then we use this finding to development a new cultivar of cotton. That is, you can express the double-stranded RNA of the pathogenic fungi as a molecular target and transfer it into the cotton. So we produce a high disease-resistant cotton. And now he is almost will be obtain the certification of transgenic gene plants in China now. And another one, do not show here, you know, in the last month, we also we found out biotechnology to defend the Huanlongbing of the orange plants, because that disease is caused by insect-borne bacteria. And there is no in Florida of America, in Southern America, and in China, this disease almost destroyed the orange industry. And we, based on the finding, because we found the regulatory of disease resistance in orange is conservative, so we designed a peptide by the machine learning and to defend the disease. So the results are published on the science, but there is many cooperation, including the Tupan and the Syngenta has meet us to discuss how to use this technology to defend the Huanlongbing in the future. And this is one of our role of the microbial resources in the synthetic biology, because you know this is an AI-driven world now. But the big data, most of big data are fake data. If you ask ChatGPT or DeepSeek, you use a run word, he will output a run result. So the data is most important, but the data have to come from the microbial resources. So based on our data, we designed a variety of enzymes for the industrial usage. This is an example. We designed a high efficiency enzyme to decrease the plastic PET. And so I think the microbial resource is more and more important because it is our humans' most important present from the nature. And secondly, it can provide big data for the AI or machine learning drive the science research in the future, including the environmental science, agricultural, medical, and the industrial. So in the future development direction, I believe with my colleagues that the golden age of microbial resource and the big data time is now here, not coming, is now here. So we will, one of our reform is to combine our two departments into one. One is a CGMCC. The other one is a National Data Center for the Microbial research that is led by the Professor Qian Wei. So now Qian Wei is the director of this new department. We want to use our collections to make big data. That big data is not only used by ourselves, it should be used by all of the scientists from our world. So this is why we participate in the ACM meeting here. And based on the resource and based on this data, we can design, for example, the enzyme. We can design the biosensor. We can design the platform to produce everything. We can design the future vaccines or drugs to defend diseases. So I think this is quite important now and in the future. And we also set up a Beijing K Lab. Beijing K Lab is use our collection and the big data to have an intelligence laboratory for synthetic biomanufacturing elements, including the transcription factors, sensors, enzymes, genome editing elements. All of these elements can use as bricks to build a new microbes as a platform. That microbe, even you do not see it in the nature, to produce our future requirements. So in a summary, I want to say the scientific advance is important, and we want the policymakers to more and more know the microbe resources is quite important, not only for basic research, but also for the development of the future of this world or the country. And before I close my talk, I want to say, because this I put in the new version, we also have assigned many or renewed MOU with various country friendship, including the Japan, Uzbekistan, Brazil. And this morning, I and the Malaysia group, we renewed our MOU. And I also visit the KCTC and KCC. All of us are friendship in science study and in the microbial resources. So I think we should frequently meet and discuss how to move the microbial resources for the future use. Thank you, and thank you very much.